Welcome to the CCCExplorer video demonstration. CCCExplorer is a Java-based software that predicts and visualizes the gene signaling network to aid research on crosstalk identification in the tumor microenvironment. CCCExplorer integrates a computational model that we developed to uncover cell-cell communication as a direct and connected network. These cell communications range from ligand-receptor interactions to transcription factors and their target genes. I will now show you how to use CCCExplorer. Here's the first window you see when you open CCCExplorer. There are three main sections. This is the cell type input section, which allows you to upload a gene expression profile under different conditions from different cell types. This is the parameters settings section, which allows you to set parameter values for a selected algorithm. And this is the results section, which displays the predicted crosstalk signaling pathways between different cell types. To run a CCCExplorer project, you have to input at least one cell type. You can add a cell type file from our remote database or from your local folders. Clicking the Add Source from Server button displays the cell type list from our server. These cell types were published in our Cell Reports paper. You can click to add them directly into the project. In the Parameters section, click on the drop-down menu in the Direction box to change the direction, or in the Method box to change the method. You can change the default parameters for both to and from cell types. Then click the Run button on the toolbar to start the project. The project will take several seconds to finish. If the run is successful, the result will be displayed in the Results section. The left view in the Results section is the crosstalk gene signaling network. The pathway table on the right contains all enriched pathways and corresponding p-values for the generated network. The bottom table includes all ligand-receptor pairs identified in the network. Clicking on one pathway highlights it. You can use the zoom buttons to scroll your mouse wheel to zoom in or out on the pathway's network. The Show Hide Edges button can be used to hide or show edges of the pathway network. This function will be helpful if there are too many edges in the network. You can select the gene node and check the gene information in the network more easily by hiding edges. When you click on the Screenshot button, CCCExplorer will take a screenshot of the current network view. Only visible genes will be saved. When you click on the Export Network button, CCCExplorer will export the whole pathway network. All genes on the pathway network will be exported no matter whether the gene is visible or not in the view. Click on Export Ligand Receptor to export the ligand receptor pairs to an Excel file. The Toggle button is used to show and hide the gene node information. If the button is on, a tip box will show up when you hover over the gene node. No tip box will appear if this button is turned off. When this toggle button is on, CCCExplorer will highlight the genes that can be targeted by one or more drugs in Drug Bank. A drug list will be displayed in the drugs table when you hover over a highlighted gene. Double-clicking on one drug redirects to the Drug Bank website, where the detailed information of the selected drug can be found. You can manually modify the layout pathway network by dragging and dropping the gene nodes. Click on the Save icon to save the current project. The default extension of the file name is .smab. You can load the saved project as follows. Launch CCCExplorer again, click Open, select the desired project file. When the project is loaded, CCCExplorer will display the last modification saved. CCCExplorer also allows you to check if a gene is predicted to be involved in crosstalk between certain cell types. Type a gene name and click Search. If the gene is identified in certain crosstalk analysis, The display information will include the cell types, underlying algorithms, and bar plot of the gene expression profile. You can apply a customized algorithm for crosstalk analysis through the Add Method function. To add a customized algorithm, you need two files. One is the main algorithm file. The other is the configuration file. The configuration file includes the names of the algorithm and the script file, as well as the list of parameters used in the algorithm. After the customized algorithm is successfully uploaded, you just need to select the customized method from the drop-down menu in the method box. From here, the steps are the same as using the default method. For more detailed information about CCCExplorer, consult the user guide. Thank you for using CCCExplorer.